record this lecture so that you could still get the content and not fall behind. Of course, I'm more than happy to answer any questions that you have when I see you in lab on Monday. We'll review just briefly the assignments that are coming up, talk about the lab, what you've been doing and where things are going. We'll back up in the content before we move forward to talk about why chemotherapies fail or the development of drug resistance. And then I wanted to see if you had any questions for the radiation oncologist that will be Skyping in next week. All right, let's start with the schedule. Next week, there's a module assignment due on Thursday at 11 a.m. As usual, you guys have a couple of days where you can be late. So you can submit it as late as Saturday, February 16th at 11 a.m. and receive a 25% deduction. But it's, like I said, always worth it to submit late. The concentration of the unknown is also the pipetting exercise. So the expectations for the pipetting exercise are that you can achieve an R-squared value of 0.983 times. And the way that you're evaluated on that is your estimation of the concentration of an unknown protein sample, the bovine serum albumin that you've been getting. That will be due on Wednesday, March 6th at noon. So that will be before your lab. And we'll have open labs on Fridays between now and then. And of course, if you need any additional time in the lab, just let me know and I can let you guys into the Python lab to work on this. We have a plate reader up there as well. Okay, so let's take a look at the lab. I want to remind you again that this experiment is based entirely on fly development. I like to frame it this way because this is the way that I understood it best when I was learning this from Tintin Sue. You've already learned how to maintain adult flies. So we made food and we poured food into the bottles, and adult flies are normally maintained in those bottles until such a time that we want to synchronize efficiently the ages of third instar larvae that we're going to collect. So in the lab so far, what you've done is maintained an adult colony of breeding flies. Those are the ones that are on the shelves in the lab. We've collected embryos from population cages. The purpose of this is to increase egg laying and also to synchronize ages. Now we can synchronize them. I've gotten as close as 1 hour. Shorter than that isn't all that successful because the females kind of take their time laying eggs. But after an hour, you can synchronize them so that they're all within an hour or 2 hours, or in our case, between we'll say 4 to 8 hours. It really depends on convenience. We'll have some plates that have to sit overnight, in which case I'll come in at 11 and collect at 7 a.m. But we're going to try really hard to synchronize them, mainly because if we're collecting the third instar larvae, after we transfer them to bottles, we're going to lose some the less synchronized they are. In other words, if we let a plate sit in the population cage for 8 hours, and then we pull the plate at the end of the 8 hours, we're going to have embryos that were just laid. So they'll be minutes old, and we have ones that are 8 hours old. When you separate the larvae, once we start to see third instar larvae, we're going to lose some because some will be too big and some will be too small. So the the least amount of time that we can keep those plates in the population cages, the better. It turns out that around 4 to 6 hours is the most efficient in my hands. After we collect the larvae, we're going to irradiate them. And you did that in class last week. And then we transfer them into vials where the developing larvae will be eating a single compound. And the idea is that we'll then be able to evaluate survival of adult flies in those vials after about a week. So here we are again. You made food to maintain adult flies in these bottles. And then we synchronized the ages in a population cage. You collected larvae from the grape juice plates. The females really like to lay eggs on the yeast paste using either a spatula or a small metal spatula or a paintbrush. I think we all use paintbrushes. You're able to liquefy that yeast paste and then pour it through what we call an egg collection basket so that the yeast paste, the liquid yeast paste, will flow through, and then we collect the embryos in the egg basket. You'll see that we use embryos and egg interchangeably in the lab, mainly because we're not sure if they're embryos yet, but in all likelihood they have been fertilized. Last week also you collected third instar larvae. You would have either poured them into a secondary container or dumped the bottles containing the third instar larvae directly into a 3-tiered sieve system, where the middle tier is going to, by diameter, the diameter of the mesh, select for third instar larvae, which you can see here. The way that I can tell that there are third instar larvae in the bottles is that you start to see this tooth, and you can see them sort of almost swimming around the top layer of the food. From there, you made vials that contain just the regular food, 5 milliliters, and in the real experiment, what we're going to do is put 5 microliters of compound in there, whether that be the negative control, positive control, chemotherapy that we know works, or the unknown compounds that we're going to be testing from the National Cancer Institute. After making the vials, you transferred somewhere between 50 and 100 larvae. I know that this blop is kind of hard to quantify when you just blop it in there. You have to visually estimate. The thing that makes it challenging is not only that there are a lot of larvae in this dish, but also that there's cornmeal. The cornmeal doesn't dissolve and it sticks to the larvae. So it is difficult to estimate. But the rationale for 50 to 100 is just that you need to have about 50 to get statistically meaningful numbers. Based on preliminary data and a statistical test called a power analysis, we know that we need to have 50 to say something significant about a population. And 100 is really just my qualitative maximum number that I'm willing to count in these vials. If you imagine more than 100 vials or 100 larvae in these vials, it's going to be very difficult because they start sitting on top of each other. Next week in the lab, we're going to start marking the genotype of these. And if you imagine taking a marker and circling them, realistically, it's not really possible to mark more than 100 on the wall. So between 50 and 100 is really quite qualitative, but also there is rationale for choosing that range of larvae into the vials. Okay. So after we're done with our practice, which this week will mark the end of the practice of the techniques required to do the drug screen, we're going to move on to the actual drug screen. But next week, after marking these vials and quantifying alive and dead flies, you're going to move on to a class where you just have pipetting. So for 2 hours, that's the only thing we're going to do. And Andy and I will walk around and help you guys work out how to get the 0.98. All right, so in terms of content, you already know that we structure the class so that you're learning techniques in the lab, and at the same time, you're learning the basis of the research, sort of the research story. So what have we talked about so far? We've talked briefly about some of the causes of cancer. We talked about the differences between risk and cause, which one of you brought up, and it was a valid point. In the literature, you'll see that it's a clear distinction. However, when you read sort of lay material, you'll see that there are statements like tobacco causes cancer. Well, does it cause cancer? It could, but a more accurate statement is that it increases the risk of developing cancer. So we made that distinction after this was presented to you. But the point here is to show you that there are lots of different causes. And in each of these cases, for very different reasons, they increase the chances of developing mutations, either through causing injury, which causes the cells to have to divide faster. And faster dividing cells, more DNA synthesis, just leads to more mutations because you have more DNA synthesis. And other things will directly damage the cells, the DNA itself. So it causes mutations directly. The hallmarks of cancer, when somebody develops cancer, are variable, but generally speaking, cancers exhibit one or more of these characteristics. So probably the most common one that we think of when we think of cancer is a cell that is able to keep dividing and dividing. So what we call that is sustained proliferative signaling. In other words, the cell is either telling itself through an abnormal way to grow, to divide, or other cells in the area are sending signals to that cell telling it to divide. Growth suppressors are a natural way for the cell to prevent its own growth, or for cell neighboring cells to say stop growing. A cancer cell often can ignore those signals and continue to grow. And as it develops more mutations, it acquires more of these hallmarks of cancer. The cells are developing a lot of skills. Some of the skills are useful, like being able to enzymatically degrade extracellular matrix and get out of the tissue and into the bloodstream. Other skills aren't going to be particularly useful for the cancer cell in terms of developing a tumor and metastasizing to distal tissues and organs. Treatments today are mostly based on the hallmarks of cancer. So the hallmarks of cancer paper that's referenced in the reading is really famous. I think it's the most, at least one of the most referenced papers ever, because it's not only the basis for identifying what a cancer is, but also for identifying how one might treat a cancer. We, in this class, are going to focus mainly on traditional chemotherapies here and radiotherapy or radiation therapy, because this is our strategy. But you should be aware that there are also other strategies like surgery, which is a very common strategy. It's been used since probably the beginning of time. You present with a tumor, and the first thing that you do in most cases is take that tumor out, unless it's not possible because the tumor is too deep in the tissue, or it's too dangerous because it's too highly vascularized, or the cancer doesn't produce a tumor at all, as in the case of leukemia, before it begins to settle in other tissues. Precision therapies are what we called in the reading targeted therapies. These are ones that are developed mainly after you've genotyped the tumor and have a sense of what is the primary cause or the biggest cause of the problem. So for example, there are targeted therapies that are developed to inhibit growth factor signaling. There are a lot of types of cancers. Lung cell carcinoma is an example. Also breast cancer is another example where growth factors are either upregulated or the receptors are upregulated, and molecules can get in and block that signaling. But keep in mind that in those cancers, even though that's the primary cause or the biggest problem, there are loads of other mutations that are contributing to the uncontrolled growth of those cells. So one strategy may be good. It may help to keep things under control enough so that you could figure out a second strategy. But one strategy rarely works in any type of cancer. Combination therapies are really a newer way of thinking about things, not only in the sense that if you use two different things, you might be more successful in getting that tumor to stop growing, but also with the realization that many tumors have cells that have mutated so much that they become resistant to the initial therapy. So if you can either prevent that or address it right away, it's a real benefit to treating cancer. So why do chemotherapies fail? We already know that some chemotherapies either don't work in some patients or stop working. So let's talk about some of the reasons why that happens. The first has to do with the way many drugs are designed. So if you can imagine developing a cancer therapeutic back when there were no treatments at all, you would already know a little bit about what the tumor does or how the tumor behaves. So you would know that the cells in the tumor are dividing rapidly, unlike in other organs where growth is more restricted. So for example, in the liver, you could lop out an entire lobe of your liver and your liver will regenerate. But your liver knows the cells signal to each other when to stop growing. The tumor doesn't. So if you were thinking about how would I stop this tumor from growing, it might make sense to target the cell cycle. So there are a lot of drugs on the market that target various stages of or phases of the cell cycle. So for example, you might target S phase. And if you had a drug that targeted S phase, you would be looking at a drug that inhibits the ability of the cell to make new DNA. And that's a great strategy because if you can't make DNA, you can't divide. Unfortunately, not all cells are in S phase at the same time. So often when people are on these drugs, you have to keep them on an IV for many hours or do repeated treatment with the hope that you'll catch all of the cells, or at least most of the cells, while they're in S phase and while the drug is still in the system. Other drugs will target things like microtubules, and in fact, the positive control that we're using, called colchicine, is a microtubule inhibitor, and that will prevent spindle formation and chromosomal separation at the end of mitosis. But again, that also requires that the cells be in the correct phase of the cell cycle. So when you're treating with these chemotherapies, the treatment has to be slightly longer term. Another reason is there might be a minimal immune response. Now, this is an interesting one because a lot of chemotherapies rely on the immune system itself for various reasons. Now, a minimal immune response might sound like a bad thing, but if you think about a maximal immune response to a tumor, then you're also talking about autoimmunity. So if you're relying on the immune system to target the cancer cell, it requires that the immune system do it in a smart way. So if the immune system can identify cells that are cancerous, or in this case, what we're showing is the response of the immune system to an infected cell, which is an appropriate response. In that case, the T cell or the immune cell will bind to the infected cell and kill it. But the system can tell that this cell has something else going on inside it because the surface markers are different. This could be a virus or even bacteria that infects the inside of the cell, and you start to see these proteins on the surface, and the immune system can recognize those things. This is normal, but what we want to do, instead of causing a normal reaction, we want it to be weaker. We want it to be what's called self-modified or modified self. So you can see a cancerous tumor might have an upregulation of, say, growth factor receptors on the outside. Then you would cause the immune system, maybe through vaccination, for example, to cause an immune response against these receptors. But you want this binding to be weak because many of the normal healthy cells are also going to have these receptors expressed on their surfaces. So this is a very tricky situation where it's very efficient because you're using your own system, the patient's own system, to target the cancer cell. But on the other hand, you're trying to modify the way the immune system works. And that's very challenging in getting a chemotherapy to work or continue to work as the system gets smarter and gets around it. A major reason why chemotherapies fail is that the tumor itself will change. So in this example, what we're showing is the original founder cell, or the original cancerous cell, that started to divide to form a small tumor. Now, the more those cells divide, the more mutations they're going to accumulate. So suddenly you're going to have this mixed population within the organ of things that look clonally. They're basically clones of the first founder cell. And then you also have the acquisition of new mutations that create almost a different phenotype, right? Because you're getting different proteins expressed in this cell, and over time you get a more and more mixed population of cells in this tumor. And in terms of a treatment, if we again think about how we might target this tumor, each of these mutations might be conferring a hallmark of cancer. So if we think about the red cells as conferring, say, the ability to increase angiogenesis, and then the purple cells to increase cell division, and the blue to decrease the ability of the cell to be sensitive to anti-growth signals, this would require 3 different treatments to be successful. This is an example from a real tumor. The primary tumor was tested such that they took punch biopsies from the tumor called R1, R2, R3, R4, and these are all in different regions of the tumor. The metastases are going to look similar to the tumor, but they might only look similar to some of the regions of the tumor. In other words, as this primary tumor starts to metastasize, it's not all of a sudden going to explode in all directions such that all of these regions are represented in the metastases. Rather, what's going to happen is you're going to get metastases that look like different regions of this tumor. So now what happens is you have a tumor that's fairly large in the origin tissue that's acquired different mutations so that it has different cellular phenotypes. And then the metastases settle in different organs, and they too look different than the original tumor. Here, this was a group that tested these different punch biopsies, the 7 of them, and then looked to see how genetically similar they were to other tumors, either within the tumor itself, the different regions, or in those metastases. So what you see on the bottom on this x-axis are the different regions, R1, R2, R3, R5, R8, and R9. So those are represented in the tumor itself. And then M1, M2A, and M2B, if we look back, are the metastases located in different organs throughout the body. The blue and the red indicate an up and down regulation of genes, where each box represents a different gene. So in this example, you have one gene all the way across, and you can see that it's doing opposite things in different types of different regions of the tumor or in different metastases. So again, in this example, what you see is this gene, the expression changes uniquely to the rest of the primary tumor, but it does seem that that part of the primary tumor is the thing that metastasized. So there are other things that are common among all of the tumors, and then there are things that are unique to all of the tumors. This is a reason why it's very, very challenging to treat these cancers because they are genetically distinct from each other. Another reason is genetic drift in the tumor after treatment. So what we're talking about in number 3 is the natural development of phenotypic instability, where the tumor itself is starting to look different from other pieces of the tumor. But you can induce this kind of phenotypic instability through treatment itself. So if you give a patient, for example, with the same kind of tumor, with a kidney tumor, a treatment, and it's mostly responsive to that treatment, the tumor will shrink, which is considered a success. So that's fantastic. If you do the same sort of genetic analysis, this is a microarray where they test many thousands of genes and look for up and down regulation of the genes. Not knowing much about genetics, you can see that the initial tumor and the tumor that remains look somewhat different. So there are some similarities in the pattern, but what's left is a tumor that was probably resistant to this initial treatment. So you got rid of the cells that are susceptible to the first treatment. So that's fantastic. You have a smaller tumor, but now you have a different tumor. So you give another treatment, or maybe you retreat with the same drug. That causes cell death and then subsequent proliferation that causes growth of the tumor. And now the tumor looks genetically completely different than the first tumor. So just said a different way, if you treat a tumor with a drug, it's going to work for a lot of cells. But then the tumor that's left has cells in there that may be resistant to the first tumor, just because they were never susceptible to begin with. You treat again, the tumor gets bigger over time, not because of the treatment, but just because it gets bigger. And then what happens is you have a genetically distinct tumor. So now, although the tumor may look the same, and it may be around the same size, this tumor here is basically a brand new type of cancer that needs to be treated in a unique way. The fifth reason that is really important to cover is drug resistance. And it's important to emphasize here that your cells are already drug resistant. Your system is set up to get rid of things that don't belong. So if you take ibuprofen, your cells are going to upregulate pumps on the surface, almost every cell in your body, to get rid of that ibuprofen because it's not supposed to be there. It makes sense. Now these pumps and channels were originally developed biologically, or at least it's thought, to resist excess amounts of things that are in the environment. So it's not that we evolved when we started getting exposed to different types of drug, but rather that system was already in place. So drug resistance can occur at many different levels. If we just look at the way that a drug is taken and what happens to it, if we start over here, you may know already that the drug is absorbed, usually through the intestines or maybe through the endothelial lining of your blood vessels. It gets distributed into many different tissues. Cells in those tissues metabolize it or break it down, and then it gets eliminated in various ways, but usually through urine. The drug then, once it gets into cells, induce a different kind of response. So this pharmacokinetics describes how the drug moves through the system, and the pharmacodynamics, or PD, describes how a cell responds to that drug. So the drug goes in and it sometimes gets activated. It may require activation by the cell, and that's exploiting the expression of enzymes that are already there. It could also result in direct cellular damage, and the cell dies. The way that the cell is able to resist these things is by inactivating the drug, and that's not a choice that the cell is making, but rather there are enzymes that are already in that cell. And as those enzymes begin to get used, it causes signaling patterns in the cell that can increase the expression or existence of those enzymes in the cell. There are other adaptive responses, and also there are ways of this that the cell can resist apoptosis so that it doesn't actually die when it gets affected by the cell. Now we're going to focus here just for a minute on alterations on the drug target, because this is one that is not appreciated, I think, as much as perhaps it should be in cancer, because cancer cells are mutating so quickly. I mentioned earlier that there are mutations that confer productive changes in the cell in terms of being cancerous. So if we go back and think about the hallmarks of cancer, some mutations can cause the cell to acquire those abilities to call more blood vessels into the tumor, to resist apoptotic signaling. But one thing that's not, I think, often ignored is that the cell itself can just by accident acquire mutations in the way that the drug interacts with its own molecules. So an example of this is growth factor signaling, where a drug may diffuse into the cell and interrupt the way that the growth factor receptors activate. So if the drug is not able to inhibit anymore because of the shape of that protein, then the cell can ignore the presence of the drug. As long as the drug accumulation in the cell isn't toxic to the cell, it can ignore the effect of the drug completely. Another way that's probably the most common way that drug resistance occurs is through drug efflux. As I said, every cell in your body has the ability to pump out actively, using ATP, drugs that get taken up by your cell, either through a pump, a channel, or diffused through the membrane. That cell is already set up to pump these drugs out. This is not a thing that occurs in response to a drug. These things are always expressed on the surface of your cells. So let's take a look at this quickly. If we imagine that this cylinder is a tumor cell, on its surface, it has a way for drugs to go into the cell. And I want to emphasize here that this is shown as a channel, but some drugs don't go through channels. Some drugs are able to diffuse right through the membranes, and others require interaction with things that are brought into the cell anyway. So keep in mind that this is just a schematic to show you drugs going in. Efflux pumps, in your reading you learned, are ATP dependent and require a conformational change. So let's see what happens when you give a chemotherapy. So the chemotherapy, through whatever mechanism, is going to get into the cell. And as it gets into the cell, it's going to start to accumulate. That cell, through efflux pumps that exist on the surface anyway, is going to start pumping out that drug because the cell is set up to protect itself. You give more drug, and ideally the drug accumulates inside of the cell and the cell dies. In some cases, though, and I would say even in response to most insults like drugs or xenobiotics or an excess of things that might damage the cell will upregulate the presence of efflux pumps on their surface. So now you give the drug goes into the cell, and then it starts pumping it out very quickly, preventing the accumulation of the drug inside of the cell. Now this happens in normal biology. I wanted to show you this. This is some work from my postdoctoral fellowship where I was interested in cardiomyocytes. I was also interested in a multi-drug resistant efflux channel called MDR1. Now this is complicated because these efflux pumps are affected by a lot of things. So if you've ever heard, for example, that if you're taking a certain medication, um, you shouldn't drink grapefruit juice. Grapefruit juice shuts down how cells metabolize drugs. So if you drink a lot of grapefruit juice, there's something in the grapefruit juice that will inhibit the ability of the cell to deal with that drug, and you'll experience increased toxicities. Now, MDR1 is an efflux pump, and I was interested in this in the context of chemotherapies and the development of cardiotoxicity. In other words, I noticed in the literature that a lot of patients who are taking these new small molecule inhibitors of growth factors are ending up in a cardiologist's office after they take the chemotherapy because they're in heart failure. So I thought, why would that be? And then furthermore, anecdotally, a few doctors were saying that they felt that women were experiencing this toxicity a little bit more than men. And in doing some research, what I saw was that women historically have always expressed to doctors that they have more toxicities. And historically, this was called hysterical woman syndrome. This term was also used for women with migraines who were taking medications to treat the migraines. So women were coming into the offices of doctors and saying, you gave me this drug, and now my kidneys hurt. Now I have headaches all the time. And the doctors were saying, oh, it's just hysterical woman syndrome. It turns out, at least in the case of these small molecule inhibitors, I observed that estrogen itself decreases MDR expression on the surface of cardiomyocytes. So what you see here are isolated cardiomyocytes. I took them out of rats. I basically degraded the extracellular matrix of the heart and separated the cardiomyocytes out and then cultured them and exposed them to a vehicle, which in this case was dimethyl sulfoxide. It's just the solvent for the chemotherapy. Now, I exposed those cells to serum relevant doses of sunitinib and many other small molecule inhibitors that inhibit growth factor receptors. And what you can see is that when the cells are exposed to a chemotherapy, expression of the efflux pump goes up. That makes sense. That's a completely normal response. Most cells will increase expression of efflux pumps like MDR when they're exposed to a drug. So that was very reassuring when I observed that. But what was interesting is my female rats and mice were both developing cardiotoxicity and heart failure at a much higher rate than males. So when I gave these cardiomyocytes estrogen, and again, this is a serum relevant dose. This is something that you'd want to watch for in the literature. Is, if you put in a ton of anything, it's going to kill the cell. It's going to cause changes. But the doses that were used here are doses that are relevant to the concentration of estrogen observed in the serum of women. So if we give estrogen to these cells, just put it in the culture media, you see a very dramatic downregulation of this MDR1. Interestingly, if you treat the cells with sunitinib, with the chemotherapy, and then expose the cells to estrogen, rather than having an upregulation of MDR1 in response to the sunitinib, there's a massive downregulation. In other words, the sunitinib, in this case, in the presence of estrogen, is not being pumped out as efficiently in these cardiomyocytes. So the conclusion of this paper was that we should watch female patients who are taking these drugs or consider putting them on a hormone blocker so that they can take them and not experience increased toxicities in other tissues. Now, some people asked, well, wouldn't this be an effective chemotherapy co-treatment? Absolutely. Because if you can imagine having cancer and you can only tolerate some dose in the serum just because the dose is so high and you can't pump that much into your serum, you might then co-treat with something that's going to decrease the ability of the cancer cells to deal with that treatment, so it accumulates in the cell. It's a perfectly valid approach. However, with chemotherapies that are taken by mouth or by IV, what you see are increased toxicities too, because there's no way to direct at this time a chemotherapy or an MDR inhibitor in cancer cells only. It affects all of the cells in the body. So we know already that cancer is caused by abnormal alterations in DNA that increases growth and metastasis, among many other things. Those alterations in DNA can also lead to mutations in the targets of drugs that can lead to drug resistance. Right now, the survival rate for all cancers, this is the 5-year survival rate, is about 68%. So chemotherapies are good, but they're not as good as they can be. They fail because of drug resistance in many cases, and these are because the drugs are specific to the cell cycle. They rely on the immune system, or the cells themselves are able to evade it through mutating the way that the tumor looks to the chemotherapy, and also the tumor cell becoming resistant through pumping the drug out. I just want to highlight one additional thing that came up last week, about a week ago, in the Jerusalem Post. This was very transiently on social media and in the news that an Israeli company had found a cure for cancer. I know I got several emails about this, and I just wanted to go through what this paper actually said. This group said that they have a cure for cancer for all cancers, one set of peptides or small proteins that will produce, in their words, a complete cure for all cancers within a year. What it's called is Mutato, or multi-target toxin. Mutato is basically a mix of different peptides that are intended to bind to the cancer cell, and they have different targets. So the idea is this is a combinatorial treatment in itself, in that it's already utilizing multiple different mechanisms to kill the cancer cell. So basically, it's a combinatorial treatment, which is a fantastic approach. So what have they done so far? They show in mice and in mouse cells that when they treat with different doses of the drug, they get increased cell death, which might not be apparent here, but I can tell you that what they're showing are cancerous cells that are going through apoptosis here. They've done it in mice, as I said, and they're going to begin clinical trials soon. The thing that I want to put into context for you, if you run across this paper, is that they did it once, and it was not published or peer-reviewed. So what they're reporting is their own internal data that hasn't been looked at by anybody else. But they claim that it's repeatable, although they themselves haven't repeated it. They also did micromolar doses, which is extremely high. To achieve micromolar anything in your blood is very, very difficult. I would argue that anything at 10 micromolar is going to be toxic. In my own studies, nanomolar and picomolar was what I was observing for chemotherapies, not just the small molecule inhibitors, but really any approach. And also, they're claiming that they'll be able to get this to people within a year. Clinical trials in this country take between 12 and 15 years, so I'm not sure how they're going to get through safety trials and efficacy trials in 1 year, but this is what they're claiming. The ACS chief medical officer is just throwing caution out there and saying, be careful, because they may very well be able to get something on the market in Israel within a year, but in the United States, we'll talk about this, the process is much longer to prevent catastrophic injury in patients. And this was seen in thalidomide, which was fast-tracked through the FDA. We'll talk about that example later on in the semester when we talk about clinical trials. But I just wanted to give you that example because you may have heard about it. And if you're interested in that paper, I can get a hold of it and email it to you. Okay, so next week, I'm not sure if she's going to be able to come to our lecture, but she's checking her schedule, but we'll be able to have a radiation oncologist Skype in, and you can ask her questions. What I'll do in the event that she can't come to our lecture is on Monday, I'll collect some questions from you so that you can ask, and I'll record her answers during the main lecture. All right, and I'll see you on Monday.